Okay, we're going to have a look at a dihybrid cross, and we're going to see is this idea of linkage, if genes are linked. Now, usually that means that the two genes, they are linked, they're on the same chromosome, and they are also pretty close to each other. We'll see why that is in a minute. Okay, but we're going to be considering a cross in Drosophila. Okay, Drosophila, small fruit fly. They're used because when you mate them, they usually have about two or 300 offspring. Between mating and the adults hatching out, it's only a week, so the experiments are pretty quicker than doing it with pea plants or whatever. Okay, so the two mutations we're going to look at are this one, which is small wings. Now the normal version is the capital W. That's the normal gene, normal size wings. Small w is the mutation which gives the small wings, and so it's a recessive gene, so you would need that to have small wings. If you're heterozygous, if the fly is heterozygous, it would have normal. Same thing with eyes. There's a mutation which causes the fly to have no eyes, and it's recessive. So for the fly to have no eyes, it would have to be small n, small n. Heterozygotes would have normal eyes. Okay, so we're going to do a cross between two double heterozygotes. So there we've got, yeah, a male and a female heterozygous for both of those genes. Okay, now, so let's think about the possible phenotypes. We'll just look at the left-hand column for the time being. Okay, so the phenotypes here. So normal wings and normal eyes, small wings and eyes, normal wings with no eyes, and then a double mutation where you've got small wings and no eyes. Okay, now you'd expect to get those phenotypes in a 9:3:3:1 ratio. We all should be familiar with that. And so let's say, have a look at this. I say just look in the left-hand column for the time being. Well, let's look at both columns if you like. So I've done that. There's two experiments there. I'm just making these results up. So, right. So each time, if you get the 9:3:3:1 ratio, you would expect 113 to be normal. Again, how do we work that out? Well, we work it out from here is 9/16 multiplied by 200. There's 200 flies altogether, is 113. Okay, so that's how I did that. Okay, you get 38 with a single mutation, 38 with the other single mutation. The double mutation, you should get 12 with that phenotype. Okay, and the same in the right-hand column. Now, what we've got in the next column here is what you actually get. Okay, so let's look at, this is one experiment on the left. Let's say somebody did it and got these results on the left. Now, we can see our numbers, they're a little bit out there, they're not perfect, but they look like really they are probably fitting into that 9:3:3:1 ratio. Whereas we look at the experiment on the right-hand side, right, these two don't look too bad, yeah, but you're getting far fewer flies with a single mutated phenotype than you'd expect. And that is probably because those two genes, if you did get these results, the two genes are close together on the same chromosome. And so in meiosis, they both tend to move the same way into the same gamete. Okay, so let's now do a chi-squared test on all of that. Right, now remember this chi-squared test, this is how you work it out, this purple box here. So you do observed O minus E, it's observed minus expected, square it, and divide by E. So you get all of these all of these purple numbers here, and then you add them all up together to give you the chi-squared value there. So add all those purple numbers up together on the left and you get 5.39. Okay. Right, now when we do it, let's have a look at the right value. Now, first of all, you notice your chi-squared value is way bigger there. The results are not fitting into the null hypothesis. The null hypothesis, remember, I'll write it up here. The null says no difference between observed and expected. Right, that is definitely not the case for this experiment down here, is it? Right, and as we say, the main problem is with these two things here. When you've got a single mutation, you're not getting enough of those. You get far fewer of those than you would expect. The numbers are a little bit down. So that means the difference between the observed and the expected is fairly big. And particularly, well, for that one, you basically got too many of the double mutation, and that contributes an awful lot to that pretty high value for the chi-squared statistic. Okay, now remember that the bigger the chi-squared statistic, the less likely those results are to, the less likely the null hypothesis to be correct with just a bit of random variation going on. So now we need to look at the probability value. So we need the chi-squared table down here. Degrees of freedom, remember that is n minus 1, so that's the minus 1. So that's the number of phenotypes in this case, which is 4 minus 1. So we will need to be looking at there's 3 degrees of freedom there. And let's look at this 5.391 first of all. Well, it would be, that would fit in about there, wouldn't it? So for probability, I'm just guessing there would be roughly 0.2 or something like that. So, or you can say it's 20%. So that means what that is saying is that 20% of the time when you do that experiment and the null hypothesis is correct, you would get results which are that far away from the expected. And that's okay. So if 1 in 5 times you do it, you get very similar results to that. So that tends to suggest the null hypothesis is correct, right? Here, well, 17.5, well, that's not, you know, that is, the probability is less than 0.01. So it's less than 1%. So if you did that experiment, that cross 100 times, you would not even expect to get results that far off. Even once in 100 times would be less than 1. So we can safely say that the null hypothesis is incorrect there. There is a significant difference between the observed and the expected. Remember the usual cutoff value we have is, we say p, the biggest value p can have is 0.05. Sorry, the smallest value it can have is 0.05. So that means 5%. So let's say we say, you know, you do it 100 times, 5% of the time you'll get the results, and that's acceptable. And so the biggest value chi can have there, 0.05, is that 7.81. So that's the cutoff value for the chi-squared. Well, this one is 5.39, so that's okay. This one is way over it. So this is, you've got the results on the right, you definitely know you're wrong. Your null hypothesis was incorrect. Right, let's have a little think about what's causing that then. So just a bit of space here. Right. Okay, so right, in this case, the null hypothesis is correct. So Mendelian rules are being followed. So that means the two genes. Remember, I had one which was the wings one. I think was the red. And in red, the wing, and the one that caused the eyeless, that was that in blue. So the two genes would be on different chromosomes, probably. Okay. So remember, at meiosis, there's one chromosome, there's another one, and you've got your let's say you've got the wingless gene, sorry, the small wings on that gene, on that chromosome, and the gene for the no eyes on that one. Right, this is its homologous pair. So I'll do it in green, and obviously the gene's going to be in the same place there and there. Right, now this is in the cell, so the homologous pairs are lined up at the equator of the cell. There's the spindles. Meiosis is going to occur. So the gamete you're going to get there is you get, okay, so I just draw it, and you get those two chromosomes there. So there's your genes. But of course, what could equally happen is, because you get independent assortment of the chromosomes, random assortment of them, so they could swap round, okay? So that chromosome, that could be a green there, that could be the black one there, in which case you get a different combination of genes in the gametes. That one would be, just cross that out. And that's going to, whichever one happens is going to be on a 50-50 basis, isn't it? So you are going to get this, the two sets of gametes in equal amounts, so you will get the 9:3 to 3:1 ratio that you would expect. Right, let's see what's happening in this one here, though. Now, in this one, you're not getting independent assortment of the genes in meiosis. That's not happening. And the reason for that is because they must be on the same chromosome. So I need to draw one chromosome here. The genes must be on the same chromosome. And not only that, they're probably fairly close to each other. So there's its homologous chromosome. So there is there's the gene for the wing. And there's the gene for the eye. They're fairly close to each other. Right, so meiosis is going to occur. There's the spindles. So you're going to get the two alleles are going to segregate together. So let's say, for example, this one was the mutated version of each one. Okay, so that's going to be that could be the mutated wing, mutated eye. So that would be small one. Yeah. And the other chromosome, the other homologous chromosome, which separates to go in the other gamete, that would have the two dominant ones, okay? So that'd be capital E, okay. So, right, so if that happened, then you would never ever get any of these ones, these two, where you get a single mutation. That would be impossible. But we do get small numbers of them, and of course the reason for that is because recombination does occur sometimes. Okay, so a recombination event, what would happen is you'd form a chiasmata here, and they'd exchange genetic materials. So you'd get that from the green chromosome, you get that from the black chromosome. Now we can see what we've done there is we have effectively swapped over the gene for the eye color. So I'm going to have big E there and a little e there. So that means that that big E can go with a little W in the gamete sometimes. Not very often, because you need, for that to happen, you need to have a crossover happening here. And you can imagine if the genes are very far apart, then there's a pretty high chance of a crossover occurring. It may happen so frequently that effectively those two alleles are sorted independently, in which case you would get results like that, and there would effectively be no linkage between those genes, even though they're on the same chromosome. They're so far away, that one there, that one there, that almost every time you're going to get a recombination event. As I say, if you get these sorts of results on the right, and the null hypothesis is incorrect, then you do have linkage. The genes, the W and E, would be said to be linked. Interestingly, also, the distance between those two genes is you can work it out basically by looking at these numbers of these two things. The more of these things you get, the further apart they are. The less of them, that one, Okay. Now, let's think about Mendel. So Mendel, why didn't he see linkage? He didn't. All of his results, he didn't see any linkage. Well, Mendel did his work on 7 different genes in pea plants. And so we now know which chromosomes they're probably on. And the genes, they're not all on different chromosomes. Some of them are on different chromosomes, but the ones that are on the same chromosomes are, the case where they're really far apart from each other. So that means you wouldn't see any linkage. So Mendel, he didn't know anything about chromosomes. He didn't observe any linkage, and so that didn't cause him any problems. Incidentally, there have been suggestions that Mendel did cheat a little bit in his results, because I think some statisticians have pointed out that they are so close to the observed value that the probability of that happening is actually quite small. But that's debatable. But the thing is, Mendel did not observe linkage.